quite the computer person. She's also fun to have around because she's very outdoorsy, and at the beginning of each of our weekly VAB meetings, we go around the table and say, hey, what'd you do last week? And I go, oh, I took an exam, and I gardened. And she's always done something interesting over the weekend. She's gone rock climbing or looking for nudibranchs on the coast or skiing or something. So it's always kind of fun to see what they did on the previous weekend. Turns out she also discovered that she likes teaching and is quite good at it. And in fact, after this quarter, she's going to be hired as an instructor to continue with the introductory biology course. And so with no further ado, let's hear about her work.
Okay. Thank you. Thank you, Mike. Can everyone hear me all right? Awesome. Alrighty. Thank you all for joining me on this beautiful afternoon. This is Host Genetics Shape the Microbiome, Insights into Snail Resistance to Parasites. Let's get started. Alrighty, here's a brief outline of everything I'm going to cover today. I'm going to start about talking about the disease I study, so what is schistosomiasis? We're going to talk about the genetics of snail parasite compatibility, 2 key genomic regions, snail microbiomes, my experimental designs, aims, and methods, my results, and then I'm going to wrap it all up with my conclusions. Alrighty, schistosomiasis is a tropical disease in humans, meaning that it's prevalent in impoverished communities in tropical regions around the world. Over 250 million people are actively infected with schistosomiasis, and there are over 700 million at risk, and it's estimated to have 200,000 annual deaths. It's second only to malaria as the most devastating parasitic disorder. It's transmitted via a trematode parasite called a schistosome that requires an aquatic snail intermediate host prior to infecting a human host. Here we can see the distribution of the different schistosoma species. I study the species outlined here in green called Schistosoma mansoni. It's prevalent in Africa and parts of Latin America. And we study one of Schistosoma mansoni's intermediate hosts called Bioflora glabrata. Alrighty, I'm going to briefly share about the schistosome life cycle. Starting in the human host, the adult parasites will sexually reproduce. Their eggs are then shed into a freshwater system, and when they come into contact with the water, they'll hatch, and then the new parasites will then search for a snail host. If they find a compatible snail host, they will infect the snail, mature, and asexually reproduce. They then exit the snail host back into the freshwater system, where they will search for their next human host so they can begin again the life cycle. Alrighty, schistosome infection causes a variety of both acute and chronic symptoms. An infection in humans can last anywhere from 3 to 10 years, with some of the longest cases lasting up to 40 years. Here we can see what the adult parasites look like inside of a vertebrate host. These adults will then mate. Their eggs will migrate to the liver in a human host. The eggs will then try to traverse out of the organ to exit the human host. And in that process, it causes severe tissue fibrosis or scarring of the organ. I'm not going to share some of the other more gnarly symptoms of schistosomiasis. If you want to do your own research, I'm going to leave that up to you. There's no vaccine for schistosomiasis to date. The WHO recommends treatment with the drug praziquantel, but the drug is limited in success as it only clears an active infection and does not prevent reinfection, and it's also difficult to administer at large scales. Recent efforts have turned towards more integrative approaches of also controlling the snail host population in addition to administering the drug. But current snail control strategies are unsustainable as they often incur a high ecological burden, such as biodiversity loss or environmental contamination. Therefore, there's a need to develop more efficient control strategies at the snail stage. There's 2 outcomes to a snail and a parasite interaction. The first outcome is when a parasite is able to evade detection by the snail host. This is a compatible interaction, and it results in a successful infection. These snails are deemed susceptible to parasite infection. The second interaction is an incompatible interaction. And in this scenario, the snails identify the invading parasite and they will destroy the parasite before it's able to mature. This leads to an unsuccessful infection, and these snails are resistant to parasite infection. Trematodes and snails have been co-evolving with each other for millions of years, and this has resulted in the parasites evolving complex counter strategies to the snail's ever-evolving defense system. This dance of reciprocal adaptive genetic change between the two hosts has resulted in substantial genetic variation in both the snail's resistance and the parasite's ability to infect the snail host. Alrighty, the snail immune response relies on a coordination of both the snail immune cells, called hemocytes, and humoral factors. Here we can see the two different interactions that I mentioned previously. On the left, we have the compatible interaction where the parasite goes undetected by the snail host. And on the right, we see the incompatible interaction where the parasite is identified by the snail host. If you notice the barrier that surrounds the parasite, that white space, those are the snail's hemocytes that are surrounding the parasite, encapsulating it, and destroying it. Ready. Revealing the network of genes that control snail parasite compatibility could lead to new ways of controlling schistosomiasis, as the allelic combinations of genes that produce a resistant phenotype could be targeted to alter natural snail populations, rendering them less susceptible to parasite infection. Today, we know of 7 genomic regions where naturally occurring allelic variation controls snail resistance to parasite infection. And we're going to focus on 2 of those regions, the Guadalupe resistance complex and the polymorphic transmembrane cluster 2. Right, so first I'm going to share about the Guadalupe resistance complex, or the GRC for short, okay? We know a lot about the proteins in that are encoded by the genes in this gene region. So I'm going to do a bit of a rapid fire round to catch you all up on everything we know about this gene region. We found that one GRC gene is expressed on hemocytes, which are the snail immune cells, and influences the number of parasites shed from infected snails. So we know it's involved in the snail schistosome response. We found that different GRC genotypes, and here when I use genotypes, it's just different versions of the GRC gene region, differed in their oxidative burst activity in response to galactose-glycans. Galactose-glycans are just a common pathogen-associated molecular pattern, and that oxidative burst activity is what we see when that encapsulation process is happening and the hemocytes are destroying the parasite. We found that different GRC genotypes differed in their whole snail microbiomes, which suggests that these proteins may be playing a role in modifying the snail microbiome. We also found that this region has a highly divergent group of single-pass transmembrane genes, or TM1 genes for short. We found that these TM1 proteins are membrane spanning and also have this hypervariable extracellular portion, which suggests that they might be interacting with pathogen-associated molecular patterns. Altogether, all of these studies are consistent with the hypothesis that the different GRC proteins are likely functioning as a membrane-bound pattern recognition receptor on hemocytes and have immune significance. All right, now I'm going to talk about the polymorphic transmembrane cluster 2, or the PTC2. Okay. We discovered this region more recently, so we don't know as much about the function of the proteins in this region yet. Here is some work that was done by my lab, and this figure shows the different PTC2 genotypes on the x-axis, and then the percentage of snails that became infected when we introduced them to parasites. We found that the S allele is dominant and confers high susceptibility to parasite infection, and the R allele is recessive and confers resistance to parasite infection. We also found this region contains 11 coding genes, and 8 of them are these transmembrane genes. And it's also highly divergent in structure and sequence. So the reason I shared so much about the GRC region is because we think that the proteins encoded by the PTC2 may be functioning in similar ways to the GRC proteins. Besides the transmembrane domains, this group of proteins or this group of genes shares no homology to the GRC region or to any other class of proteins to our knowledge. So yeah, these findings in conclusion, are all also consistent with the hypothesis that the PTC2 proteins may also be functioning as a sort of pattern recognition receptor that has immune significance. Okay, so in addition to these immune recognition proteins, the snail microbiome is speculated to play a critical but unexplored role in the snail schistosome interaction. Microbiomes are a set of microorganisms and their genes that are specific to a site or to an organism. And Biomphoria glabrata actually possess this open circulatory system that is filled with hemolymph. Hemolymph is just the equivalent of circulatory fluid or blood in snails. And from the literature, we know that the snail hemolymph has similar microbiome diversity to the whole snail microbiome, and that it's also distinct from the external water microenvironment. Pathogens that enter the snail host into the hemolymph tissue will encounter both the snail immune defense cells and the symbiotic microbes that are occupying the shared space. Because we found that different GRC genotypes had different microbiomes, I want to ask that question today of do the different PTC2 genotypes have different microbiomes? And I want to focus in specifically on the hemolymph tissue. Alrighty, so I just covered a lot of information, so let's do a real quick recap to make sure we're all on the same page, okay? I studied the snail Biomphoria glabrata, that's the intermediate host for the Schistosoma mannsoni parasite. These snails naturally vary in how resistant they are to parasite infection, and we're focusing on two regions that are known to be associated with that resistance to parasite infection. We found different GRC genotypes had different microbiomes, so I want to ask the same question of do the different PTC2 genotypes have different microbiomes in their hemolymph tissue? Alrighty, onto my experimental design, aims, and my methods. In this study, I worked with snails that varied in how resistant they were to parasite infection. These snails came from multiple inbred lines of a population of Biomphoria glabrata snails that my lab developed by undergoing 2 to 3 rounds of selfing. That resulted in inbred lines that are fixed for either the RR resistant PTC2 genotype or the SS susceptible PTC2 genotype. Each of the inbred lines is housed in their own snail tank and has their own water. And the hoses that we see supplying these tanks are aerating the tanks, they're not water hoses. When we challenge these snails in these snail lines to determine their percent susceptibility, we found that the snails from the SS inbred line, shown in orange, were nearly 100% susceptible to parasite infection across the board, versus the snails from the RR inbred lines were a bit more variable in their response, but overall were more resistant to parasite infection. Using these inbred lines, I was able to go on and sample 8 of the RR fixed genotype inbred lines. And from each of these inbred line tanks, I randomly selected 4 replicate snails and 1 water sample. I also sampled 10 of the SS fixed genotype inbred lines and again captured 4 replicate snails and 1 water sample. I also sampled 1 bonus line that was still segregating at the PTC2 genotype. That means that it contains all 3 of the PTC2 genotypes, so PTC2 locus. So it contains all 3 of the PTC2 genotypes, RR, RS, and SS. This line is called the 15RS inbred line, and I sampled 30 snails from this line. After I genotyped the snails, I found that I had randomly selected 9 SS snails, 10 RR, and 11 RS snails. Great, and now I'm going to share my experimental aims for this study. My first aim was to just describe the microbial biodiversity of the snail hemolymph tissue and of the tank water microbiome. So I wanted to see if the tank of the snail microbiome is just simply reflective of its tank water microbiome or if it passively acquires it. For my second aim, I wanted to test whether the snails fixed at the PTC2 locus for either the RR resistant genotype or the SS susceptible genotypes differed in their hemolymph microbiomes. Then I wanted to test if the tank water that was housing those snails also differed in their water microbiomes across the RR or the SS genotypes. And then finally, I wanted to test if the snails that were still segregating at the PTC2 locus and in that 15 RS tank and sharing a common tank environment differed in their hemolymph microbiome compositions. Alrighty, so now it's time to start the experiment. I went into the snail room. I collected 4 replicate snails from each of my inbred lines. I also collected my water samples at this time. I size matched the snails to make sure they were all about the same age. I then extracted the hemolymph from my snails via the heart puncture method, and then I immediately froze the hemolymph in the water on liquid nitrogen to preserve the microbiome. Next step was to generate my microbial data. I began by extracting and quantifying the microbial DNA. I then sent that DNA to the Center for Quality of Life Sciences core facilities at OSU and to undergo 16S rRNA amplicon sequencing by focusing on the V4 hypervariable region. And this was done on the Illumina MiSeq platform. And we did this following the Earth Microbiome Project protocol. I then did all my data processing, visualization, and analyses in R. All right, now let's check out what I found. To take a first look at our microbiome compositions, I created these composition bar plots. So here I have all of the hemolymph samples shown. Each bar represents an individual snail's microbiome makeup, and then the inbred line that snail belongs to is plotted on the bottom. On the y-axis, we have the abundance of those bacterial taxa at the phylum level. And our big takeaway from this plot was just that there seemed to be a lot of variability across the snail individuals in terms of their microbial makeup. Next, we took a quick look at the water microbiome samples. And yeah, again, we saw that across the different samples, there seemed to be quite a bit of variability in terms of their microbiomes. Yeah, there was a microbiome present in the water. Next, I analyzed alpha diversity of my microbiome samples. Alpha diversity is a measure of biodiversity within a sample, okay? So it looks at both bacterial richness and the evenness of the abundance of those bacteria. Here I have alpha diversity quantified as effective species using the Shannon entropy index shown on the y-axis. And then I have my different treatment comparisons shown on the x-axis. I have the water in the hemolymph from the SS inbred line shown in blue, and the water in the hemolymph shown from the RR inbred line shown in red. And what we saw is that regardless of their genotype, the snail hemolymph had a higher alpha diversity than the water that housed them, which led us conclude that the snail hemolymph has a more diverse microbiome composition than its tank water. Next, I analyzed beta diversity of my microbiome samples. Beta diversity is now making comparisons across samples. Okay, and to do this, I did principal components analysis, or PCA. PCA is a statistical technique that reduces dimensionality in complex data sets while preserving as much variability in the data as possible. The points that I have plotted up here right now are all of my water and my hemolymph samples. And the points that are closer together are the samples that have more similar microbiome compositions versus the ones that are farther apart have less similar microbiome compositions. So if the microbiome compositions of the two groups are different, we will start to see clear groups forming versus if they are similar, we'll see the groups overlap among the points. And if I change the shape of the points based off of what sample type they are, we can see that the tank water forms a clear group in the upper right. And the hemolymph samples also have their own group. They have more variability in their microbiome composition as well. I can additionally change the color of the points to reflect their PTC2 genotype so that we can make comparisons across the RR and the SS PTC2 genotypes. And then finally, I can add on ellipses that represent the 95% confidence intervals of the group centroids. My group centroids here are hemolymph or water. So therefore, we can make the conclusion that the hemolymph and the water groups do have different microbiome compositions from each other. And this was supported in our Permanova analysis. We found our Permanova p-value was highly significant. So for AIM-1, we found the snail hemolymph has a diverse microbiome distinct from the surrounding tank water microbiome. This suggests that bioflavonoid glabratus snails are likely actively managing their microbiome versus just passively acquiring it from the external tank water microenvironment. Next, I did principal components analysis on the hemolymph in the fixed genotype inbred line snail samples. The points are colored based off their PTC2 genotype. So I have the RR resistant snails shown in red and the SS susceptible snails shown in blue. We can see some groups forming between the two groups. There's partial overlap here, but there's two groups. Our Permanova analysis found the genotype effect was highly significant. So we therefore do make the conclusion that the different PTC2 genotypes do have different hemolymph microbiome compositions. We also noted in our Permanova analysis that the inbred line variable had considerable among line variation as the R squared value was 0.45. This was not surprising though, as the inbred line variable reflects both the common tank environment that those snails are sharing within their inbred line and also the genetic background of the starter snail that was used to propagate that inbred line. The other thing we noted on this PCA was that the SS snails exhibited much more variability in their microbiome compositions compared to the RR snails. I wanted to explore this further. So what I did was separate PCAs on the RR snails and then again on the SS snails. Here, all the points are colored based off of what inbred line those snails belong to. So in the RRPCA, we can see that the 4 snails that I sampled from each inbred line seem to group really near their inbred line mates, such as like what you see in the 6RR group. Those 4 red points are really close to each other. But when we jump down to the SSPCA, we don't see that same pattern repeating. The points from the same inbred line seem to be all over the place. They jump around, right? So not only did the RR snails exhibit less variability overall in their hemolymph microbiome compositions, but they're also more similar among their inbred line mates as well. Next, I wanted to ask the question of what taxa are responsible for this genotype effect. Okay, so what bacterial taxa are very different between the resistant and the susceptible snails? To do this, I use Bayesian models to analyze microbial abundance and then again, microbial presence or absence. And I found that the microbial abundance of 61 taxa significantly differed between RR and SS hemolymph microbiomes. And then in terms of presence absence, I found that 27 taxa significantly differed between RR and SS hemolymph microbiomes. Here's a look at one of those taxa, just so we can get a visualization of what's going on here. I have abundance of that taxa on the y-axis, and then my RR versus SS snails. Alrighty, and that brings us back to our second aim. We found that the PTC2 genotypes have different hemolymph microbiomes in the fixed genotype inbred line snails. We made this conclusion based off the Permanova analysis, the PCA analysis, and also that taxon level analysis that we conducted. This suggests that the PTC2 proteins may be playing a role in modifying the snail hemolymph microbiome. We also observed that RR snails have not only more similar microbiome compositions to each other, but also more similar microbiome compositions among their tank mates. This suggests that the RR snails may be exerting different constraints over their microbiome compositions than the SS snails. That results in them having a more uniform and conserved microbial community structure as opposed to the SS snails. Next, I did principal components analysis on the tank water samples that were housing those fixed genotype inbred line snails. And we again colored the points based off of what PTC2 genotype they were, and we do again see two groups that form. Our Permanova analysis found that the genotype effect was highly significant again. So we make the conclusion that the tank water housing the fixed genotype inbred line snails also differed in their microbiomes, which is interesting. And I'll have some more notes on this later in the talk. All right, next I did principal components analysis again on the 15RS snail hemolymph. Okay, so recall that these snails are still segregating at their PTC2 genotype, and they all share a common tank environment. So it's coming from one tank. We saw that regardless of genotype, they had very similar microbiome compositions. There was no genotype effect going on here, okay? We were a bit surprised at first to see this result after seeing such a strong result in the fixed genotype inbred line snails. Therefore, we proposed two hypotheses to explain this difference in results. Our first one is a hypothesis of reciprocal influence, okay? What we think might be going on here is that the tank water microbiome may be strongly influencing the hemolymph microbiome and vice versa. So in the RR or SS fixed genotype inbred lines, there may be sort of a positive feedback loop that is happening between the snail microbiome and the snail water microbiome that reinforces the genotype effect. And this permits the genotype-specific microbiomes to be detected. Versus in the shared 15RS tank, the presence of all three PTC2 genotypes in that tank may have resulted in a more homogenized microbiome composition that prevented the genotype-specific composition to be detected. This may also explain why we saw a significant PTC2 genotype effect in the snail tank water PCA. Alternatively, what could be going on here is that there is no genotype effect, but instead the effect we're seeing is due to an experimental artifact. Here's a picture of what our snail tanks look like in our snail room. We have all of the RR tanks on one rack and all of the SS tanks on another rack. These racks are sitting right next to each other in the snail room. They're touching each other, so that top right tank in the RR rack is basically touching the top left tank in the SS rack. Ideally, we would have alternated the tanks and put them every other, but because they're on separate racks, we can't exclude the possibility that they may have experienced different microclimates between them that cause this rack effect. Although in the snail room, we did keep these tanks temperature and light controlled. We kept them on a monitored air system. We also fed the snails similar amounts and kept them on similar cleaning schedules. So we're not sure what kind of experimental variable could have caused this effect. The other thing I want to note here is the location of the 15RS snail tank. It's on the RR rack, and it's the farthest away from the SS rack that it could be. Ready, on this next PCA, what we first see here are the fixed genotype hemolymph snail samples shown. We notice that the RR samples are clustering towards the bottom left, and the SS samples have more variability, but sort of tend to cluster more towards the right. If the rack effects hypothesis were true, we would expect the 15RS samples to have more similar microbiome compositions to the RR tanks because they are on the RR rack and less similar to the SS tanks. But when we add on the 15RS samples, we see that they actually have more similar microbiome compositions to the SS snails. So this effect argues against rack effects as an explanation for this effect we're seeing. That brings us back to our final aim of we found that the PTC2 genotypes did not have different hemolymph microbiomes in the segregating genotype inbred line snails. As we have strong evidence against the rack effects hypothesis, we think that what is going on here is the reciprocal influence of the snail hemolymph microbiome on the snail water microbiome and vice versa. Ready, and now let's move on to the conclusions. So yeah, we did all of these cool studies, all these cool findings. So what does it all mean, right? First conclusion is, well, we found in this study that the PTC2 resistant and susceptible genotypes have different hemolymph microbiomes in the fixed genotype inbred lines. We also identified various bacterial taxa that are highly associated with the resistant or the susceptible snails. Our first conclusion is that host genetics definitely play a role in shaping the hemolymph microbiome composition in these snails, and further allelic variation in this single locus caused the snails to have different microbiome compositions. Helps us to further understand how these PTC2 proteins may be functioning in these snails. It's also the second study to show that these highly polymorphic gene regions enriched with transmembrane genes have different microbiomes, which helps us to understand how these important resistance loci may be acting in these snails. We also found evidence that the water may be a crucial mediator of the snail microbial communities, and future work may further explore if this effect is, or to what extent this effect occurs. These findings also raise the interesting possibility that these immune loci may be affecting resistance to schistosomes indirectly through the snail microbiome versus through direct contact with the invading schistosomes. This means we might be able to affect snail fitness by manipulating the microbiome. Altogether, these findings help us to further understand how the snails and the schistosomes are interacting, and it brings us one step closer to mitigating the spread of human schistosomiasis. Alrighty, now I'm going to switch gears a little bit. I would like to take a moment to thank everyone who helped me throughout this process of getting this degree. My first thank you goes out to the members of the Bluin Lab, past and present. Thank you, Mike, Stephanie, Javier, Shun, all of our undergrads, Anna Gabriel as well. Thank you all so much for all of the assistance throughout the years. I could not have done this without you. I really appreciate it. Thank you also to all my committee members. And like Mike said, you really helped me out with all of the microbiome analysis, and I learned a lot, and it ended up being one of my favorite parts about this project. I really appreciate the time that you spent with me. Thank you to Virginia, who's watching in from Zoom, for all of your helpful and insightful feedback over my project. Thank you to Devlin for being my GCR. I really appreciate your help throughout this process. Also, thank you to all the folks in the integrative biology department. It's clear today that we have a really fantastic department who really shows a lot of support to everybody. And I'm really thankful to be a part of this department. Next, I want to say thank you to the beautiful friends that I have made here during my time in Oregon. You've made life really fun here. You've all brought so much joy and laughter into my life. I simply could not have done this all without you guys. Thank you, Kyle, for all of the support, all of the encouragement. I really couldn't have done this without you. Um, you bring, I'm so thankful that you came into my life, and I could not have asked for a better climbing partner to go throughout life with. Also, shout out to my family who are all listening back home right now. You all are my true day ones. You've been there since the very beginning. Um, Shout out to my little sisters, Chloe and Livy. If you're still paying attention at this point, I'm impressed. Thank you for reminding me how my love of biology began. And yeah, I hope I can use this degree to make this world a better place for you too, and to my little niece Harper as well. I want to give one final shout out to Oregon. From the snowy Cascade Peaks to the rocky intertidal zones on the coast, Oregon's been perhaps my biggest form of emotional support throughout this process. Um, the nature here has wowed me, and I'm really thankful to have gotten to explore the diversity of life that is shown in this state. So yeah, thank you to Oregon. And with that, I will take any questions. Felipe, yes, thank you.
One quick question. I was struck by the variation amongst our lines and their susceptibility. And there's a early preliminary plot of the challenge.
Yeah,
There's a lot of variation there.
yeah.
Some of our line genotypes are still very susceptible.
Mm-hmm.
So it seems like you have the opportunity there, assuming you sequence across that range of phenotypes in the RR.
Mm-hmm.
You have the possibility to test for how the different taxa are parsing out across genotypes. You could, there's simple regression type thing that you can see the impact of the different taxa, proportions of different taxa
Mm-hmm.
as you susceptible RR genotypes.
Yeah, yeah, for sure.
I don't know if it makes sense.
It does, yeah, yeah. We do that, but it would be an interesting thing to look at. Yeah, yeah, for sure. Yeah, Chini.
Wait, well, I guess bigger picture, I was wondering if you have an RR in the mix tank that it shows this, I wonder if it shows the same level of susceptibility that you have the same RR genotype differences in the microbiome. How that would relate to susceptibility.
I think I'm a little confused by the question.
In the fixed tank, and they have a very
Yeah. Mm-hmm.
Separate microbiome.
Yeah. Mm-hmm.
I wonder what would be the outcomes for those two genotypes.
Mm-hmm. In terms of susceptibility, yeah, if we went on to challenge those snails, like if they would be maybe more susceptible in the RS-15 tank, yeah, because they were more susceptible. Yeah, that's a really interesting question and definitely a possibility, right? We don't know if the snails, because the SS snails had more variability in their microbiome compositions, right? Maybe that could be playing into that susceptible phenotype. We don't know if that's true, but it's definitely an interesting thing to look at in the future. Yeah, Holly.
I was wondering, how would these findings compare to snails in the wild? What would you expect to see?
Yeah, good question. Yeah, so these findings aren't directly transferable, right? Because these snails are from an inbred line population of snails. They underwent 2 to 3 rounds of selfing. So that means that they are still segregating at anywhere from 25 to 13% of their genome is still segregating. Okay. Um, what these snail lines allow us to do is study the average effects of the PTC2 region at the RR or the SS susceptible genotypes, and then go into the wild snail populations of snail and look at how this loci is acting and see or have an idea of where to look in the first place when we go into the wild snails. Mm-hmm.
Right, well, so what'll happen next is Jade and her committee will go back to Quarterly Hall and we'll chat for a while. And then when we're done with that, I'll send out an email and invite everybody to the 4th floor for snacks and toast.
Yay.
And so that, I'm guessing that'll happen probably around 4 o'clock. All right, thanks for coming.
Yay, thank you so much. Thank you.